NWMMD 2022. Let's start this occasion by stating a quote. The sky is not the limit, and the sky has no limit. So is the knowledge of young minds, which has no limit to gain. So let us all use this platform to gain knowledge. Now, I would like to welcome Dr. M.D. Balakumaran Sir, Head in Charge, Department of Biotechnology, TDGDVC, to address the gathering.
Thank you, Karthika. Respected
Thank you,
Secretary
sir.
G, Principal Dr. S. Santosh Babu Sir, our Chief Dr. Geena G. Pillai, Founder, Jasa Innovation, Bengaluru, Karnataka, distinguished speaker, head and faculty from other departments, participants from various institutions, and my dear students. On behalf of Department of Biotechnology, Dwaraka and Goverdhan Vaishnav College, it is my great pleasure and honor to welcome you all for the national level virtual workshop on molecular modeling and docking. I deem it a great privilege to express a sincere gratitude to our beloved Secretary, Sri Ashok Kumar Mudraji, for giving us this wonderful opportunity. With great privilege, I welcome our Principal, Dr. S. Santosh Babu Sir, for this inaugural session. I would like to take this opportunity to express my sincere thanks to our beloved Principal, who has always supported us at our every initiative to make it successful. We are honored with the presence of our Chief, Dr. Geena G. Pillai, Founder, Jasa Innovation, Bengaluru, Karnataka, who has a good experience in the field of machine learning, molecular modeling, software, mechanistic driven drug discovery. I extend a very warm welcome to all the renowned speakers of the national workshop. I extend my warm welcome to the department head and faculty members from various other departments who are present here. Computational techniques are an integral core of biomolecular research. Software applications provide fundamental support to experimental studies in biochemistry, biotechnology, and bioinformatics. Pharmaceutical companies and the agriculture sector rely on designing molecules through high throughput screening, docking, and molecular dynamic simulation. Computational drug discovery methods provide a valuable insight into the understanding of protein-ligand interaction systems in a visual manner. The last few years have seen a rapid development of computer-aided drug design methods, which have reached a level of accuracy that allow routine application in the field of drug discovery. Ladies and gentlemen, I sincerely hope the today's workshop would help explore and expand the knowledge in the field of molecular modeling and docking, and offer a great platform to have a fruitful discussion in the field of bioinformatics. On behalf of the management, students, and staff, I once again extend a very warm welcome. Thank you. Jai Hind.
Thank you so much, sir. Now, I'd like to welcome Dr. M. R. M. Shivashankaran, sir, Organizing Secretary, NWMMD 2022, to give a short introduction about the workshop theme.
Good morning to everyone here. It gives me a great pleasure to explain the theme of today's workshop. Bioinformatics is an interdisciplinary science that develops the methods and software tools for analyzing the biological data and has been used for in silico analysis of biological queries. Bioinformatics has become an essential component of many fields of biology on a more integrative level. It aids in the analysis and cataloging of biological pathways and networks, which are essential components of system biology. It aids in the simulation and modeling of DNA, RNA, proteins, and biomolecular interaction in structural biology. In the post-genomic era, the rapid increase in the number of sequences of known proteins witnessed the availability of possible drug targets. A large number of methods and tools have been developed which provide the means to exploit the wealth of new informatics in drug discovery approaches. The molecular modeling encompasses all theoretical and computational methods for modeling or simulating the behavior of molecules. The docking is now used to help the rationalize the ligand activity towards a target of interest and to perform the structure-based virtual screening campaign. Much like it was when it was first developed, it can be used at various stages of drug design process. The workshop aims to use the combination of theoretical and practical sessions in order for the participants to gain the practical experience. The objective of the workshop is to provide the hands-on training on various bioinformatics approaches such as biological data retrieval, protein sequence analysis, in silico molecular modeling, and protein ligand interaction analysis through online tools and offline softwares. I hope that all the participants makes the most of this session. The practical knowledge gained through this workshop will help them in enhancing the skills in utilizing the tools for the application in bioinformatics research. Once again, thank you all. Have a fun and enjoyable learning experience. Good day. Thank you.
Thank you so much, sir. Now, I would like to welcome Dr. M. Pavitra ma'am, Faculty, Department of Biotechnology, DDGDVC, to introduce our honorable guest, Dr. Girinath, sir.
Morning, one and all. It is a great pleasure for me to introduce our young and eminent speaker, Dr. Girinath G. Pillai, Director and Founder of Sastra Innovations, Bangalore, visiting scientist at Nairo Research, India. Dr. Pillai is one of the coordinators for Government of India and MHRD's Drug Discovery Hackathon 2020. Formerly, he was a Marie Curie Research Fellow at Molkot Limited, Estonia, and he carried out his PhD at University of Florida with Professor Katriski and Professor Carlson, University of Tartu. He holds about 40 research papers in peer-reviewed journals, international journals, and an US patent, and co-authored a book chapter on mosquito repellents. His expertise is in the area of computational medicinal chemistry, mechanistic-driven drug discovery, and modeling software. His workflows attracted global projects and ExxonMobil, the Johnson & Johnson, Eastman, US, etc. And currently, he focuses in the area of aging and longevity studies. Apart from scientific positions, he is also a mentor at IIT Kottayam, Government of India's Niti Aayog ATL schools, and research advisor to Pondicherry University, VIT University, and other institutes across the country. I am extremely happy and proud to introduce you for this session, sir. Please enrich the young minds with your knowledge. Thank you.
Thank you so much.
Thank you, ma'am. Dr. Girinath, sir, now the session is yours.
Thank you so much. Thank you everyone for warm welcome, as well as giving a good introduction about the theme of this 2-day sessions and other things. So I would like to thank Shobha Madam, as well as HOD and the coordinators, convenience, and the team who are organizing this, including the principal. And to all the participants, good morning to everyone. Just before I start, I would like to know what is your expectations from this attending this course? I mean, sessions basically. Are you experimentalist? I know most of them are students and faculties. So just wanted to understand what is your expectations? Do you want to implement it on your project, or just additional add-on learning, or you still have some challenges working on this area and you wanted to try it out, what new updates are coming up? So if you are not able to unmute and speak, you can always put it in the chat. I'll be asking a lot of questions to make it more interactive. I'll be waiting for 1-2 minutes. If nobody responds, I'll continue. So feel free to interact. If you don't understand, please feel free to ask me. It's more of a discussion rather than just talking science and going on. Now, I want to see what is your opinion, because as I forgot his name, sir mentioned molecular docking as it started, and now the perspectives have changed. Docking is still the same, but how people have looked at it and how people have implemented it the data that is being generated from docking, that has completely changed. So the perspectives we need to understand. So if you have anything like that, please go ahead and unmute and please share or put it in the chat. Okay. Yeah, someone said they want to have insights on this topic. I'm sure that I'll be able to deliver some of them today in my session. Any other? There are around 80 to 80 participants. Only one person has responded. Okay.
Good morning, sir. This is Muhammad
Yeah.
Navees from VIT University.
Good
Sir,
morning, Mohammed. Yep.
yeah. Actually, I planned actually, I'm a fresher to bioinformatics. So,
Okay.
my objective is based on bioinformatics, sir. So, I
Okay.
have to learn this. Actually, I'm learning
So you're more looking at coding or on the application part?
application part, sir.
Absolutely fine. Great. Thank you. So
Thank you,
Vijay
sir.
Lakshmi wanted to implement these techniques on her studies or projects. Absolutely fine. And can all of you tell me you're mostly from biotech, chemistry or biochemistry or bioinformatics? Apart from these, whatever I told, are there any other stream of participants here? Okay, so mainly Sonit wants to know about docking. So tomorrow there is also docking hands-on from morning. So yeah, I'm sure you'll be getting to that. So apart from biotech, biochemistry, chemistry, and bioinformatics, okay, life sciences also. Anyway, you are on board. Other than that, any other stream participants here? No. Okay, so whatever I'm going to talk, you all will understand because it's more of a life science and biology or even drug related studies. Um, okay, primer designing. Unfortunately, I won't be talking too much about primers anyway, but yeah, related areas. Oh, microbiology. Okay, nice. Okay, great. So thank you so much for that interaction. I hope more people will come in between. So let me straight away go to my presentation. So throughout my presentation, if any question comes up, if anything for yes, you can type 1 in your chat box. Anything for no, it will be number 2, so that it's easy for you to respond, right? So moving forward. So today we will look into the role of computational studies in drug discovery. We would have heard a lot, read a lot, but the problem here is still we use only molecular docking and we think that just molecular docking is everything. I don't know why. Maybe back in 2001, 2002, it's okay, but now things have changed a lot. Or just doing a QSAR is
Excuse me,
computational
sir.
studies in drug discovery.
Sorry to interrupt, sir. Your
Sure.
screen is not visible, sir.
Oh, thank you for letting me know. Let
Yeah.
me try to share it again. Just a second. How about now?
Yes, sir.
Yep.
Okay, great.
Thank you.
Thank you so much. So we still use just QSR or just docking. Uh, yeah, we always say, I want to just to do basics or I want to just to do introduction. I'm a newcomer, so I want to just learn. But all this just has to be thrown away because drug discovery doesn't know that you're a beginner. Drug discovery doesn't know that you want to just include some basics. No, drug discovery needs to be complete. We have to start from somewhere. It's good to start from all these techniques, but make sure that translation is very important. Right now, the faculties will be aware getting funding from funding agency is a biggest difficult job right now. Earlier days, it was a little bit easier, but not now. They're more looking at translational research. Earlier days, the outcome was publications, but nowadays the outcome is it's not just publication, but patent, commercializable products, or transfer of knowledge. I mean technology. This is what they are looking at. So we also have to proactively be ready with what we can deliver. right? So in drug discovery, it's not just looking at IC50 also, like you do docking and then go with IC50. Just IC50 doesn't talk about effect of the drug or efficacy. It just talks about the potency. So it's more things has to be looked at in the right manner, right? So that's what we are going to discuss today. And the great and the grand challenge for all of us is to how best incorporate existing knowledge. We learned in that way. Of course, I also learned in that way. We take an existing published paper and then try to learn, go through it, and try to implement the same methodology, making some optimizations, and then we do the same research, maybe with a different target, a different compound. No wrong in doing that for learning as a beginner, but maybe for one time, but not the next time. We keep on doing the wrong mistakes. We don't know really they have done a mistake, but we still repeat the same thing. One wrong strategy, quickly I can tell you, is that usage of filters, how to screen molecules from a natural compound database or other chemical databases. Sometimes we use a filter, well-known filter known as Lipinski rule of 5. I'll tell you what is Lipinski rule of 5. But using that at a very early stage is a wrong strategy. And that's what I said. Everybody used, if I'm also using without knowing what are the challenges there, it's a big mistake that you're, or a big wrong strategy that you're taking care. So it's something that you have to look at, is that how incorporating the existing knowledge, you have to benchmark it, analyze it, and then only implement it. They simply don't, yeah, I saw a paper in Nature, so I will just implement what they have done. No, there are papers retracted in Nature too. So make sure that we benchmark the methodologies and techniques and then see which one suits well with the project and go ahead, right? So make sure that you analyze the project data before getting involved into it. This slide is especially for students. Do not confuse between what you like and what you find easy. Basically, most of the life science students, if I ask you, can anyone, few of you quickly tell me without wasting much time, why did you choose life science, biotech, biochemistry, biotechnology, microbiology? Quickly, why did you choose? It's, or maybe how many of you think that it's just because you don't want to learn mathematics or you found mathematics is difficult? I'm putting an answer for you. How many of you thought in that way? If it is yes, number one, no, number two. Are we all connected? Oh, yeah, there are someone very honestly said one. Okay, great. So thank you so much. That's exactly what I'm trying to tell you. You found something very difficult. It's just because you didn't understand. If something is difficult for me, it's because I didn't understand. It's not that that subject or that topic is difficult. No, it's because the way I am trying to understand, I didn't do it properly, right? We cannot blame anyone else. It's the way I understood it. But I might be liking, for example, I like too much of how to, the technologies in cell biology, but I never tried to understand it. So I found it's not to be easy for me. That doesn't mean that I don't like it. So the correlation is what I am good at it, that is what you find it easy. Why I want to mention here is most of the students comes to me, so can you give me an easy project for internship? Can you tell me easy software that can work, where I can work for drug discovery? There is nothing easy unless you understand things. In order to understand, you need to read. In order to read, you need to have interest, and that's where you have to like. So make sure that you understand things to make things much more easier. So what I'm good at is what I find easy. It's not that what I like. You always like doing many things, but you end up doing what you really understand. Sometimes because of circumstances, you might do what you like, but later automatically you'll come to what you wanted to do and what you understood properly. So it's very important, students, you to decide where you want to be, at least when you're trying to do your project in the final year or final semester. Otherwise, it's going to be very difficult. I did my MSc bioinformatics, but when I did my project, I decided I have to switch my area to chemistry, basically computational chemistry and drug discovery. Because when you're going for drug discovery, more of chemistry factors come in together, just not only bioinformatics. So you have to take that turning point and decide where you have to go further. Okay, so just to talk about the company from where I'm coming. So we represent a few softwares which is used for ADME, human toxicity profiling, computational medicinal chemistry, molecular docking, including water desolvation, metallurgy, cheminformatics, CAD, ELN, and many more. We do provide internships for 2 students in a year after a drastic selection or screening process. It will be completely free if you are staying with us more than 6 months. Anything less than 6 months is charged. You might think, why the longer period is not charged? Because you will be staying with us for longer and you will be learning so much, very independent. Being for a short time, we have to be supporting you a lot. And that's why less than 6 is always charged, and more than 6, it's completely free and you will be learning a lot. So tomorrow, Devika will be covering up CSAR, which is a new perspective of molecular docking studies that could be looked at. And today I'll be overall covering the drug discovery part. Now I know many of you will be working on medicinal plants or herbals or natural products. And let's see some of the folklore quickly. But when you talk about drugs, we cannot stop not talking about chemistry also, even though you are a biotechnologist or life science or anything. Just doing docking doesn't bring the drug out anyway. Keep that in mind. So the attempt to design and synthesize a medicine or a pharmaceutical agent which will benefit humanity, such a compound could be called as a drug, right? But that involves synthesis. You have to make modification. Don't think that just simply analyzing many things on your just on the database find you a drug. Might be for repurposing. Then structure activity relationship, receptor interactions, as well as ADME toxicity studies has to be looked at. When you look at the big pharma picture, there are many compounds being rejected and very few being approved every year by US FDA. And they are called as new chemical entities. Now the question is, a lot of investments being made on all these compound research, but why only very few getting approved, where there is an estimated cost of 1 to 1.5 billion US dollars. So the amount invested on this research to get a drug molecule in the market is huge, and the failure or attrition rate is also huge. That's where we have to see how this cost is effective. I know you would have seen this slide in plenty of other presentations, but here I want to give a different perspective on what I wanted to put forward. A drug development process contains developed time, number of subjects used in the studies, total cost, and the candidate molecule. Now, even when it gets into a clinical trial, we start with 20 molecules or 10 to 20 molecules with the preclinical also. But when you try to do in vitro studies or bioassay studies or activity studies, do you use 20 compounds or you end up using 1 or 2 compounds? The biggest challenge here you might be blaming is cost or resource facilities. But unfortunately, drug discovery need not to know about your money limitations. You need to look at a much wider space. For starting, as a disclaimer, I have to always say, for a starting point, it's always good. But in reality, I wanted to tell that you should always do bioassay studies at least on 20 to 30 molecules, depending upon the funding availability, or be ready with how you have to get the funding for that. Otherwise, you don't have the room for optimization. Even if you're a chemist, you're a biologist, you're a microbiologist, or whoever, it's being relevant to everyone. If you end up with just 2 molecules or maybe 5 molecules and you say this one is the best, it's because the other hundreds you did not test it. That's why among the 5 you found the best. It doesn't mean that that is the only best available. You explored only 5. Among the 5, that was one of them among the best. So we cannot claim in a paper saying that this is the best among the many of the scaffolds or compounds available in the databases. So this is where we are looking at how we can transform our academic basic research to a level of preclinical trials, right? Which we can collaborate and do it. So this is the main take-home message from this slide. So make sure that you start doing your studies not with 1 or 2 molecules, but at least with 20 to 30 molecules, even from the docking results also. Many of the time after doing docking, we select 1 compound, 2 compound. No, please try to do at least 100 compounds or 50 compounds, and further filtering should help you to reduce it to 5 to 10 or 100 or 20 molecules, again depending upon the money you have for in vitro and in vivo studies. As we progress with the different phases, we can see the number of compounds from 10 to 20 reduced to 5 to 10, but the cost has increased. The number of years also passed on. And here you can see the funnel is reducing, but the funnel for the cost is increasing, and the number of subjects being used for testing these compounds are also slowly increasing. Population studies, effectiveness, toxicity studies all comes into the picture. At the end of the day, when we are getting into the final phase, we have a compound with 1 to 2 molecules, and then already 1.1 billion pounds being already spent, but we have already gone through 1,000 to 3,000 volunteers being tested with these molecules. With the disease and without the disease, both will be tested. And at the end, at the final stage, we get the medicines get approved with the final one. But you can see on the top, the research still continues, because after market research is very important for medicines that get approved. Some medicines get revoked, and they have to look at the side effects and many more. So it's a still ongoing process. But in our case, we end up with the publication, and what next happens next? Does someone someone took it up and move forward, or are we taking it up and moving forward? So this is what the reality we have to look at when we always work on drug discovery projects. If you have any questions, please feel free to put it on the chat. Now let us look at the role of each expertise in drug discovery phases. The first and foremost role is target identification. It's very, very important. Earlier days, many people talked about it, but many were not serious about it. When I was learning about docking studies and all, to be honest with you, I never looked into target identification and validation. I just, based on literature, tried to select the PDB and start doing docking that from the day one. No, target identification, you need to read a lot of papers, a lot of analysis. It takes few days for you to confirm which is the protein target that you have to really work on for that specific disease. And for this, it cannot be done by a chemist. High chemistry friends here, please don't feel bad. To be honest, we have to work together with different expertise to make drug discovery much more successful. So target identification is mainly for life sciences, biotechnology, and especially biochemists, and with the support of bioinformaticians, gives you a good variance of understanding the target identification. So let me give you some examples, techniques that recently we have seen in some publications. Polypharmacology, network pharmacology, where we try to implement systems biology approach, trying to identify extracted or isolated compounds from the plants, or newly synthesized molecules to generate a network of the target genes as well as the interacting partners, and also look at the disease network, where we try to understand how these forms and end up with the possible targets that we can explore for that particular ligand. Or we look at the gene card and understand how these diseases are related to specific genes. And when we try to understand for which gene we have the 3D structure of proteins or expressed ones, and based on that, we choose the target. So this is a mandatory step that we have to take care. And we also have to look at the druggability, then the whether interacting with the key amino acids, whether it is going to upregulate the mechanism or downregulate the mechanism. Hope you remember during my introduction, it was said mechanistic driven drug discovery. That mechanistic driven is something very important. Many of us forget that. We talk about it, but we forget. So what I'm trying to say here is that before doing docking, for example, if you're working on enzymes and all, you need to know what is a catalytic site, which are the key amino acids that you have to really interact with. Based on that only you can say whether your ligand or your compound is facilitating or inhibiting, whether it is agonist or antagonist. Software will not tell you. So a biochemical understanding about the target and its receptor or the role in its metabolic pathway, it's very trivial to do or understand before drug discovery. So make sure chemistry friends collaborate with the biologist to understand what could be the possible targets, not simply depending upon the literature. Then what is the research that you have done? You did a review, but rather you have to do a research to identify the target, right? So this is something that you have to really focus from now onwards. I'm sure many of you might be already doing it, but I want to give a little bit more pressure there so that you focus a little bit more on the validation part. And then we look at the lead discovery and optimization, which we already have heard a lot on molecular docking, pharmacophore, QSAR, and many more. But you can see something else also in the list, like multi-parameter optimization, combinatorial chemistry, and finding out the secondary metabolites, whether the compound is going to be stable or it is going to break down and create issues. I'll talk about it a bit later. And then we look at the drug development where we go start with preclinical tests like ADME, toxicity, PBPK, and simulations. Now, let me tell you, everybody heard about ADME. It's about absorption, distribution, metabolism, and excretion. Everybody has predicted ADME, absolutely fine. But simply predicting the ADME properties in a table and publishing doesn't give any take-home message to the reader. We really need to know the structure activity relationship. Until unless we understand the structure activity relationship, we cannot make use of the ADME predictions, which I'll talk about it in a moment. And then we will help into clinical trials. Fortunately, all of these techniques now has computational support, so computer aided design is possible. But as a disclaimer, don't think that any of these computational approaches are replacing your wet lab experiments. Never think about it. Since I don't have money, I don't have resources, I will claim that using docking water, I find it's a tomorrow's drug molecule. No, never. Even I also thought like you when I was doing my master's and bachelor's, but now after having got into a lot of failures, understanding from experiences, I have to admit that you have to validate any of your predicted computational predicted data. You have to experimentally validate it. I still don't agree that wet lab validations are more accurate but still reliable than computational predictions. Now it's a question time. When we go as a civilian, when they go to the drugstore or a medical store, they basically concern about main two things. One is how affordable is the medicines. If they're having a continuous ailment, they have to continuously take the frequency of the dosages much more frequent than a normal fever. Then they always think that. The second part they think is it going to cause any side effects or any other issues if I take this medicines regularly? But as a scientist, what do you think? What is the role of a scientist? Just tell me 2 points. What is the role of a scientist in this particular thing? As a civilian or as a normal people, they think about the 2 points that I told you. But as a scientist, what you might think are the 2 points? You can unmute or you can put it on the chat. Please make it quick. We have many questions to cover.
Good morning, sir.
Good morning.
Yeah, we would think that how effective the drug is going to be and how it is going to be the half life, how it is going to be
Ah,
degraded away from the body and not to be too much toxic to the person.
absolutely fine. But Vijay Lakshmi, whatever you said is absolutely right. The question here is, or most of the time we do this one by one, right? We filter one by one.
Yeah,
But at the end of the day, do we get a best compound?
it's a probability. It
Ah,
depends.
it depends. That's what. So we expect to get the best one, but 99% we don't get that. So that's a challenge here, which we have to see how computation methods can help. Thank you so much for that answer. Any
Thank
other perspectives? Think. Attending a session is should make you think. So brainstorming is something very important. That's where you learn. So either interact. So don't worry about your language barrier. Tamil எனக்கு புரியும் பேசுவேன், but yeah, I don't know about if there are other state people also. So don't keep language as a barrier to discuss. You can feel free in any language to discuss. Yeah, someone else can help me if I don't understand. So no other perspectives. Okay, no more time to wait. Moving forward, the two main objectives of a scientist in a general way. Okay, I'll come to drug a bit later. In a general way, the main two objectives of a scientist is that how you can explore unexplored things, or you can find an answer for unanswered question. The second role of a scientist is how to make big things in low cost. Very important. The main role of a scientist for the humanity is how to make big things in low cost. I'll tell you a quick example, nothing related to drug discovery or anything. Elon Musk, yeah, NASA rejected his project several times about SpaceX. He claimed that the rocket can go and come back to Earth. So you're saving millions of money there. But NASA didn't agree to it and they didn't believe to it. He did a lot of testing, failed a lot, miserably failed, a lot of money gone. But finally, he succeeded. Now, NASA is working together with him for SpaceX to go to space and come back without wasting a lot of money, what NASA was being doing for the past decades. So the scientist's role is to reduce the cost of medicine or reduce the cost what they are trying to explore to do big things. That was a very big example, best example I could give. So this is the same way a scientist should think, right? But in order to do that, we have to do a lot of research. We have to do a lot of brainstorming, how we can reduce that. So here also exactly this slide says how we can reduce the cost of the medicine. I'm not going to ask this question to you. We can ask maybe later. It's too early to ask that question here. But let's see why the cost of the drugs are increasing. That is because of the failures. If you look at the history, between 1960s and 1980s, the failures of oral drug molecules were mainly because of poor pharmacokinetics, that is ADME properties, and efficacy, the effect of drugs on the diseased person or a human. It was very poor. Because of these, they were being failing much more. And if you want to read much more about it, you can read get from these two review papers, which is from 1988 and 97. Later, the cause of failure in 2000 and 2003 was mainly because of toxicity and strategy. Toxicity is the biggest challenge now, because we were able to overcome the challenges of ADME properties and others just using QSAR or computational methods. We were able to reduce the cause of failure by ADME or pharmacokinetic properties. But when coming to toxicity, we didn't have too much of data for a knowledge base to come up with the predictive toxicity, especially human toxicity. Strategy is a big challenge here because we do a mix and match about techniques and pipelines, and then we mess it up. That's what is happening, which I told you. We are always trying to incorporate existing knowledge. So we have to be very careful which is the best strategy and benchmark accordingly to get the best out of it. And that's where, in order to do all this kinds of analysis or activity relationship or predicting the activity or property or toxicity of newly designed molecules or new databases or extracted or isolated compounds from plants, we have to see how we can look at it or how we can predict it, how we can analyze it. That's where the role of statistics and machine learning comes into the picture. Can you all tell me, I hope all of you are having curriculum to learn statistics for sure, right? Anybody's not learning statistics? Yes. If you're learning, yes is one. If no, two. Because statistics is something very important in any field, right? So make sure that, oh, okay, if you don't have, try to take some Coursera courses or edX online courses. You need not to pay any penny. It's completely free. If you want to take a certificate only, you need to pay. Why we need a certificate? Just learn it. And also you can access SWAYAM. our UGC, SWAYAM, and NPTEL, where you can take some statistics courses to understand, because it's very, very important in computational studies. Okay, that's where we need to understand why we need to capture the knowledge computationally, because different viewpoints, drug discovery is a multidisciplinary field, so we need to look at the different viewpoints, and knowledge transfer is something very important. Even within our own field, we have very limited experience. I would say I'm daily learning. I've still yet to learn many things. Whatever I learned might be obsolete maybe after 2 3 years. I still have to keep on updating myself. Limited memory. I would have read few papers last week, but not necessarily that I know I remember each and every sentences from there. So computers can remember or memorize much more than us. Of course, I didn't say computers are much intelligent than humans, but still the data manipulations and storing and retrieval is much, much smarter than humans. And computers have virtually unlimited capacity and perfect recall. Now, again, in another question time, uh, as you all know, there might be a difference between knowledge and information. So, what is the difference, and what do you mean by knowledge and information? Boring with too many questions? What is the difference between knowledge and information? Thank you, Aishwarya. Any other perspectives? People are putting in the chat, so if you are not able to speak, absolutely fine. That's all? No other responses? Oh my God, almost 75 people are here and only one person is responding. Okay, I'll move forward. I don't want to take too much of your time. So, as exactly Aishwarya said, knowledge and information is kind of the data or what you learn. And when you apply this data or make it use or experience it, that's where we call it as a knowledge. So knowledge is like a paint. Um, when you see a red color paint in a tin, that is called information. And when that is applied on a canvas, we try to admire, we try to realize what is the color, and that's where we understand where it is being applied. And that's where it is called as a knowledge. So computers can apply stored knowledge much faster than a person. Even scientists have hidden biases. We choose which knowledge to believe. That's exactly when I'm asking question, you're a bit biased. Which answer is correct? This guy is going on keeping and asking question. I know the answer, but I'm confused what perspective of question he's asking. So that is a common thinking of a scientist, basically. So you're all good. So computers can explore ideas more quickly and rigorously than a person. That's the reason why we're looking at the computational methods in the drug discovery. So learning denotes changes in a system that enables a system to do the same task more efficiently the next time. This explanation by Herbert Simon talks about learning. This is exactly what we do talk about pedagogy. Like we sit in the class, we learn, we do an exam, understand, then we know that we are not doing good at there, then we try to learn again, and next exam we might perform well, maybe the other way around. So this is called also machine learning. So they try to learn and understand and then try to optimize, and next time they work more efficiently. Similarly, when you do docking, when you do lead optimization, when you perform on making changes in a chemical structure, you need to understand its activity, its toxicity, and then make changes accordingly by understanding where the issue is. I also wanted to give a clear picture here. Don't think that a person need to know Python for learning machine learning. No, you don't require zero Python. What you require is a good understanding about math and statistics. If you do not know statistics, learning machine learning has no use, especially on our domain, whatever domain you are. So make sure that you know statistics and math properly, getting into machine learning. And also, you have to know certain softwares where you can apply machine learning. As I said, Python is one, but don't think that only with Python you can do. There are other platforms like Nime, Orange, RapidMiner. These are all click a button and go. You don't need to do any coding there in order to do any math or, machine learning or statistics. When you're coming to data science and AI, it's a bigger picture. Let's don't talk about that because we don't have enough data to do AI, but we are good with machine learning. How machine learning could help in drug discovery? We can identify disease prediction. We can do structural analysis. We can do drug repurposing. We can repurpose the drugs. That's the way the perspective of docking also has changed. Novel drug development can be also used. And that's where we have to see the evolution of CAD, computer-aided drug discovery. If you can see here, we start with the basic research, drug discovery, and preclinical. Unfortunately, we are all still in the basic research. In US, in Europe, I'm not comparing regionally, but still I want to tell you the fact we have to have some goosebumps to start working towards that. In US, any of the academic research, they translate from basic to drug discovery to preclinical, and then they collaborate with or they spin off with a company and do an IND submission, and then they get into clinical trials. Academic research, not companies. So in India also it's happening. I don't say no, but very, very minimal. And it's started very recently. So it's always have a good thought, plan your PhD project accordingly so that you can live with it and die with it. What I mean is you spend a lot of time to design what is your project. Make sure that the compounds that you're designing can be tomorrow patented and that can be translated to commercial. I know a student who is working on a pharmacy college as a PhD student. He patented 2 molecules, spent money from his own pocket, almost 8 lakhs for doing some certain studies and all. And I told him that that's a good investment that you have done. That 8 lakhs, what he's invested, he got a patent. Tomorrow, if he did all those exercises of data that to be shown, if some pharma companies come, he can sell it to them and he's going to earn crores of rupees. So that's translation methodology that we have to follow. So make sure that any of your research, that proposal that you're doing, not just for report keeping, but see that what is the future of that project. That is something very, very important. I'll give you a quick example. In my master's, I did something on malaria. And when I came to PhD, I did something on mosquito repellents. They're both related, right? And when I looked at some of my postdocs as well as my industrial job, a little bit of crop protection was involved. So I'm going with it along, but still on the sideline, drug discovery and other projects are also being involved. So that's how we have to plan our future if you are trying to stay within this field. I think this I have already covered. Sorry for the repetition. If you're asking for the success stories of CAD on pharma, there are more than 112 compounds based upon computational drug discovery or computer-aided drug design we were able to find. Don't underestimate or don't think that these compounds were being predicted by a software. Just one, hey, this is a compound, go for it. No, nothing like that. These compounds were one among the 1,000 molecules that was suggested by software or screened by software. So don't think that it is just one compound came out from the software, because I don't want to give a wrong message here. So these were, let's say 50 compounds were being predicted by the software. One among the 50 was these compounds. And then they were gone with the wet lab, in vivo, in vitro, all those animal studies. And then it came and at the end of the day with one single compound. So I used to do a podcast. You can search online for drug discovery research and Giri Bio, or put my name, you will be finding the podcast. So I'm talking about how computational studies have been applied on certain drugs which is already in the market. So the success stories of computational drug discovery. It's an audio, so it's only 10, 12 minutes, a quick wrap up you can get. Now, if you look at the objectives of drug discovery, I see some, I heard that some chemist students also here. So the role of chemistry is mainly synthesizing compounds, trying to see how much yield you can get, what is a new step or pathways that you're looking at, green chemistry. But the role of a computational modeler or a bioinformatician is basically, I want to predict something, good energy, good activity, a good conformation, and design some new molecules. But these guys always fight each other, the modelers as well as the chemist. Modelers come up with a new molecule and ask the chemist, can you synthesize? They will say, no, it's very difficult to synthesize it. And they give a comment, you can design anything on computers, but don't think that it can be synthesized, right? And also when coming to chemists, they synthesize the molecules just focusing on purity, etc., etc. And then they give it to modeler saying that, can you do docking for me? Now the modeler will ask, okay, what docking? Did you design these molecules based upon a disease in mind, a target in mind, or did you use any pharmacophore of existing molecule to design these molecules? The answer is no, most of the time. They say, no, based on the literature study, we designed the molecules. Then the modeler thinks, how do I then identify which target I have to use for my docking studies? So this is where there is a lot of disagreements comes between these two fields. But this can be solved where they work together. So when you write the proposal itself, make sure the chemist, the biologist, modelers work together to come to a virtual result, which is double checked, where computationally you're checking, because before doing project, you cannot do anything, rather computational studies. And then analyze together as a biologist, target identification as a chemist to see which compound to be synthesized, and as a modeler to give a predictive discovery on what all the being assumed or hypothesized, and then do a refinements, and then the proposal gets approved and you go for synthesis and all those things. In biologist, most of the case, sometimes you might not be collaborating with a chemist, but you might try buying the soft compounds. So you try to screen the database and then you ultimately go and buy the compounds. Or many people will be working on phytochemicals. So you extract or isolate the compounds and then you try to see how these compounds can be put on to use with specific targets. So now we need to have a better situation where chemists will understand what the modelers could give, and the modelers will understand what are the possible compounds that can be synthesized by a chemist. And these all can be done using computational methods. So, so we have to ask the right question to the computational studies, and right question to chemists, and right question to biologists, as well as the modelers. Don't ask the same question you ask in a wet lab in vitro experiment, the same question to a computational study. No, they are different, right? And that's where back in 1957, Arnold Moore was trying to discover drugs by blinding himself. And then he's trying to mix and match different chemicals to see whether he can come up with a new formulation. That's exactly what we're trying to do here for de novo or fragment-based drug discovery. When you're looking at the drug discovery objectives, we need to have a balance of different properties. Another question time. Oh, there are a lot of responses came in. Thank you so much, Mahalakshmi, Vaishnav, then Venkat and Amjad. So another question is that when do you call a compound to be a drug molecule? When do you call a compound to be a drug molecule? Any thoughts? When there's an action in specific to a disease. Okay. Yep, got that. Show efficacy towards a target. Okay. Any other answers? If it is bioactive. Okay. When it blocks a disease-causing agent. Manjunath, as I told you, there are drugs which are facilitating as well as inhibiting. So it's not that the drugs are always blocking, but they might also facilitate. So there are both stages. Molecules with therapeutic effect. Yeah, therapeutic is the right terminology after preclinical trials. Yes, Sharanya, that is absolutely right. So thank you so much. A lot of answers then. So once you have getting into clinical trials, then we call them as a drug candidate. Once it has efficacy, of course, when that's where the FDA filing goes in, and we call them as a drug candidate. Then why do we use in our projects drug designing? We're not designing a drug at all. We are designing a candidate. That's why I asked you that question. So when you really know that when it enters a clinical trial, when it has a therapeutic effect, all the by doing IC50, we really didn't know that it has a therapeutic effect yet. We just looked at the potency studies. That's it. So make sure that your title of the project also should match with what you're really doing it. I have done a mistake in my master's saying that drug discovery. No, I didn't discover a drug. Now let's understand what is a hit molecule and a lead molecule. Hit is a molecule, not a molecule, a group or a collection of molecules which have good potency. Any molecules having good potency, good binding affinity, or even after docking studies, they are called hit molecules. They are only called hit molecules. They are not one, they are not two, there are many, maybe 20 or 50 or 100 molecules. Hit cannot be a one molecule because that's where we are doing wrong in our strategy. Now, hit to lead transition. The hit to lead transition is because first and foremost property requirement for drug discovery is that activity, potency. When you find that a compound is active, and then the next step is to see that whether this compound can reach the target safely. It might pass through different metabolic pathways, permeability, absorption. Does it excrete at the right time? All these things has to be explored. So that's where we try to say that heat transforms to lead. In order to call a chemical compound to be a lead molecule, the compound should be potent, it should be safe, toxicity should be optimal, it should have a good absorption rate in the human intestine, the metabolic stability because it passes through liver metabolism, P450. There are different isoforms of P4, cytochrome P450. Then we have to see whether it forms breakdown, and we want to see the active form and inactive form of the compound. If it reaches the target in an inactive form, then there's no point of doing docking, right? So we need to understand that. Solubility, permeability, half-life, retention time, all those things has to be looked at in order to call them a hit molecule to be a lead molecule. Once if we have found some issues, okay, there's some toxicity, then we have to make modifications in the structure. That's what we call it as lead optimization. So you have to identify compounds with an optimal balance of all these properties, and we have to also quickly identify such situations when that is not possible. Now you might think, so come on, I'm doing BSc or I'm doing MSc. How can I do all these things? Yes, you can do computationally. Only thing you have to learn it. That's where the next question comes in quickly. But before I continue, organizers, could you please tell me how much more time I have? I think I'm already at 10:30.
Sir, you have another 20 minutes, sir. 15 to 20
Okay,
minutes.
okay, great. Thank you so much.
Okay, sir.
Uh, so we need to understand which region of the molecule is important. The biologist doesn't care about it, right? Why should I know about where the problem with the structure? Because that is the role of a chemist. No, it's not the role of a chemist. It's a role of both of you. So that's where the question comes. Next question comes. There's a compound A and there's a compound B. The compound A is very active as well as toxic at its particular concentration. Compound B is moderately active and no toxicity. I repeat, A is very active and it's very toxic at the lower concentration. B, moderately active but no toxicity. Which compound would you prefer? A or B? Okay. Many of them said A. Oh, sorry, B. Now, what is the reason? Why did you choose compound B and why did you choose compound A? I need to know the reason. It's not multi, what to say, the multi-option questions, MCQs, but you need to know the reason. Why did you choose B and why did you choose A? Very important. Most of the people who have chosen B, I presume you're all biologists. And the person who chosen A, I presume might be related to chemistry or pharmacy. Be non-toxic and effective, no toxic because it's not only no toxic. Okay, all of you talked about toxicity. Okay, Venkat, can you tell me why you chose A? No answers. Okay, fine, no problem. I'll help you with that. Thanks for those answers. Now, I told you in the previous slide, hit, identifying hit is the most important property for a drug discovery. That itself has the answer there. So activity is very important for us. Personally, I would go with A because activity is something which I'm looking at. But then it has a challenge, it has toxicity. So that I need to know where I have to make modifications to reduce toxicity. So that's where lead optimization comes into the picture. But in order to do that, I need to be a chemist or I need to have a chemist collaborator. That's the reason why I said most of the people who said B is because biologist. The main reason is even though you have low activity, you choose because of no toxicity. Absolutely fine, no problem. But when you have a compound with good activity, if you don't check the possibility of optimization, if the optimization failed, yeah, you can think of B. Now there is an argument, just for the namesake of argument, you can also ask, why can't we optimize B to improve the activity? Absolutely fine. So that's where we need to understand difference between pharmacophore and toxicophore. Pharmacophore are the different regions of a compound which prone to be active, which looks at the activity, bioactivity. Toxicophore talks about the regions which are prone to toxicity. So when you're making modifications in the structure, that is called bioisostere replacement. Bioisostere replacement. If you're trying to improve the activity, but if you're still not sure whether the toxicity will increase, there's no point of doing the optimization. So when you found a compound is more active, where you need not to change or make modifications on your pharmacophore, rather you might be modifying few functional groups or side chains, which is acceptable, and that's much more easier and practical. So the first and foremost compound that you can choose is A to optimize them. If that fails, you can go with B. And biologists mostly go with B because of toxicity. You're okay with intermediate or less activity, right? But ultimately, we need to have good activity. So in order to do that, there are structure activity relationship, so that from the structure, we will be able to understand which region of the molecule is toxic, which region is of the molecule prone to be pharmacophore, and which region has to be modified. And this helps a chemist to learn that. And similarly, that's where we try to understand what is structure activity relationship. But before that, let me, okay, this thing came in between. So target identifications can be done using systems biology approach, systems pharmacology approach. By using cytoscape, you will be able to generate a network like this, and you will be able to explore. That itself is a 4 hours of class. I don't want to get into that, so I'm just skipping them. And about the SAR, structure activity relationship, you have the experimental biological activity data. You will have the structural information of the chemical compounds, and you try to calculate certain molecular properties, and you try to correlate the activity value and the chemical structure values using statistics, which is empirical, or now we call it as machine learning. That's what we call it as structure activity relationship. Mainly, we can put it to regulatory purpose. That is where we look at QSAR. We try to take a compound, we try to calculate the descriptors or extract structural properties. Example, number of atoms, number of carbon atoms, number of oxygen atoms, molecular weight, flexibility, number of rotatable bonds. These are very basic molecular properties. There are many advanced properties. So there are more than 3,000 descriptors available. And you try to correlate between the experimental activity data and the molecular structure properties using statistical and machine learning methods like regression, partial least squares, and other methods to see how far they are correlating. And then we try to validate them, and we look at the property and activity. And this is what we call it as QSAR. But keep in mind, any QSAR model that you are generating or any QSAR model that you are using to predict should have an error value, standard deviation or standard error. Without an error value, no QSAR models or no QSAR predictions can be validated or justified. And that's where it is talking about accessibility or applicability domain. The number of compounds in the training set should also have similarity with the compounds that you're trying to test also. If that doesn't fit in within the chemical class or similarity of the structures within the chemical space, then the reliability of the prediction is poor. And that's where we look at the ADME, where we can predict certain ADME properties from the 2D structures by extracting physiochemical properties. And these are the different ADME properties available, like aqueous solubility, partition coefficient, dissociation constant, then Hurg liability, blood-brain barrier, human intestinal absorption, plasma glycoprotein, 2D6, which is isoform of cytochrome P450, plasma protein binding category, and many other physiochemical properties and thermodynamic properties. Predictive toxicology can also be predicted by the QSAR method, and knowledge-based method are where direct nexus is being using. So based upon the human clinical trial, they will be able to identify which region of the molecule is already being reported as toxic. So they give an argumental understanding which tells you that this particular region of the compound is carcinogenic. And that's where A compound can be modified. When you know that A is toxic, the chemist or your friends needs to know which region of the compound is toxic. Until unless you know which region of the compound is toxic, you cannot make the modification. So there are computational approaches where they can actually help you to understand which region of the molecule you have to make modifications. These are the 42 human toxicity endpoints available. I'm sure you would have learned about cardiotoxicity, genotoxicity, mutagenicity, skin irritations, but there are many much more which is taken from human clinical trial data. And this comes from LASA UK, and they have a consortium with pharmaceutical industry and clinical trial FDA. Then we have P450, which is we are talking about metabolic stability, understanding which region of the molecule is prone to breakdown. So if you see here, the carbon, the 2-methyl, the carbon which is close to the nitrogen, which is having C2 91%, it can break down. And by using quantum chemical calculations computationally, we were able to understand there are possible breakage that could form or metabolite that could form for a specific isoforms. So that's all from me for today. This is something that can be covered later. I don't want to talk too much here, but yeah, this is what I was trying to tell you. When I think Vijay Lakshmi was trying to tell you, try to do one by one the analysis, but at the end of the day, you expect that you can get the best molecule, but unfortunately, you might not be getting. You might end up with a bad molecule also. So make sure that you try to combine all these properties or filtering criteria together by using multi-parameter optimization. And also, finally, I want to tell you Lipinski rule of 5, and I will end the session. So Lipinski rule of 5 is a filter which talks about oral bioavailability. Even though it said rule of 5, it is not having 5 rules, it's only having 4 rules, but it's a factor of 5. What it's trying to tell is any compounds having less than 500 molecular weight and partition coefficient less than 5, hydrogen bond donor and acceptors, where donor is less than 5 and acceptors less than 10, then we say this is Lipinski rule of 5. This talks about bioavailability. Bioavailability relates to the permeability as well as absorption rate. So that's how it is related. Now, don't think that any compounds with less than 500 compounds are only accepted. And that's a wrong assumption. There are compounds in the market. There are peptides, biologics. There are even larger, small organic molecules which are having molecular weight more than 500. And this is the reason why I said this is a wrong strategy when you filter your database just based upon molecular weight. But if your protein can accommodate only a lesser molecular weight, absolutely fine. But if your protein can accommodate more, still try to explore. You can use Lipinski rule of 5 at a later stage to understand the bioavailability rather than just to filter the database. The problem, what happens here is you're missing certain compounds. You're not considering certain compounds in your screening process. If you're not considering what I told you earlier, you choose only 5 compounds for testing. From the 5, you got one best, but you cannot claim that that is the best overall because you did not explore all of them. So try to avoid missed opportunities by implementing multi-parameter optimization, which is already there since 2010. So it's nothing new, but the problem is we have not ever used it. So that's all from me. If you have any other questions, I'm happy to take it. Regarding the docking studies and others, there will be more sessions coming from Shobha Madam as well as from Devika tomorrow. So I'm not going to cover that due to the interest of time. These are some of the research outcome from my group where we have used whatever the techniques that I have told you already and widely being cited and accepted. That's all from me for today. Thank you so much for being very interactive and also sharing your perspectives and views. If you want to get the slides, whatever I presented today, you can go to slideshare.net/giribio. You will get much more slide decks there. And if you want to work on molecular dynamics, because after molecular docking, something is required is molecular dynamics. Being it's a bit difficult, people never go into it, but it's something changing. We have to work on it. So if you want to work, do molecular dynamics on your mobile phone or on a browser, you can go to my GitHub and you can use Google Colab. If you have a Google Gmail account, you will be able to run GROMACS as well as NMD in the Google Colab. It will not take any of your RAM, CPU. It will be using Google's RAM and CPU. It is free with certain limitation, but for people who are learning, it's fair enough. And GROMACS has acknowledged the notebook that I have created already. And NAMD, protein ligand MD, people who want to work MD simulations only on Windows, not on Linux, you can use NAMD. I have a tutorial video available in my YouTube channel. So just some free resources that you can make use of learning and getting into this role. Don't think that I don't have good computers. Don't worry, you have a solution here. And don't think that I don't know Linux, how do I start working? No, you have solutions for Windows too. So that's what it is. And that's all from me. Thank you so much.
Participants, any questions? Okay.
No questions? Okay, fine. I think I have to ask questions for them to answer, but rather they're not asking me questions. Okay, fine. Yeah.
Thank you for an informative session, sir. We have got to learn about drug discovery and various ideas on drug development. Participants, we will have 15 minutes break and join the session by 11. And those who didn't fill the certificate form, kindly fill it. Thank you.
Sir, Giri sir, thank you, sir. Sir, can you come on? Hello, sir.
Yeah.
Sir, can you come on video mode for a screenshot?
Okay.
Yes, sir.
Just a second. Let me check if video is working. Yeah, I think video is working. I hope you
Yes,
can
sir.
see me. Yeah.
Yes, sir. Thank you, sir.
Thank you. So bye everyone. Enjoy your next few sessions and next 2 days. I hope you learn a lot, but make sure you try this. Don't leave it for tomorrow. If you're doing something tomorrow, make sure that tomorrow itself you try, or at least during the weekends. If you don't try and keep it for tomorrow, you will never do it. So don't keep it for tomorrow. Try to at least look into it quickly, and then. So, madam, we can get you some licenses for CSAR. So you can share it with all the participants today, so they will be ready with the sessions for tomorrow.
Yes, sir. Definitely will. I'll share, sir. Okay,
Thank
sir.
you. Thank you, madam.
Oh,
Thank
thank
you,
you, sir.
everyone.
Thank you, sir.
Have a
Thanks
good
a
day.
lot for the session, sir.